Hello everyone, it's my pleasure to introduce the paper submitted to iMeta. The title is Novel Insights into Immunopathogenesis and Crucial Biomarkers Between Primary Open-Angle Glaucoma and Systemic Lupus Erythematosus. Glaucoma is the leading cause of irreversible blindness in the world, which is closely related to immune inflammatory activities. Recent studies have shown that patients with systemic lupus erythematosus have a significantly increased risk of developing glaucoma. However, the mechanism of this association remains unclear. This study reveals a new common mechanism between SLE and POAG, which provides scientific direction and basis for future research and treatment of SLE-related POAG. Our study has the following 4 innovations, including the association of new immune mechanisms between the two, the identification of biomarkers, and the preliminary prediction of targeted drugs. In this study, 384 and 1455 differentially expressed genes were identified from TM cells of POAG patients and blood samples of SLE patients by GEO. Subsequently, WGCNA was used to identify the most relevant gene expression modules with POAG and SLE. Finally, 10 key genes were obtained and their correlation was explored. In order to further explore the potential biological functions of the key genes of the two diseases, KEGG enrichment analysis and Go enrichment analysis were performed on the key genes. The results showed that these key genes were enriched in several inflammatory and immune pathways. In order to further identify the core genes, we selected each of the 10 genes based on lasso regression and SVMRFE algorithm. The genes screened by the 2 machine learning algorithms were intermixed to identify the final 5 core genes. ROC curve analysis of the diagnostic efficacy of the 5 core genes in POAG and SLE showed that the AUC values of the 5 core genes were relatively good, suggesting their potential as common diagnostic biomarkers for the disease. Furthermore, an in vitro model of POAG was constructed and the biomarkers were validated. Meanwhile, we also performed immune infiltration analysis on the disease data of POAG and SLE patients. In these two diseases, the infiltrating abundance of immune cells such as macrophages, monocytes, and T cells was high, which may also suggest that there is a certain correlation between the immune pathogenesis of POAG and SLE. We analyzed the correlation between the 5 core genes and immune cells, indicating that these key genes are likely to play a role in regulating immune-related biological activities. To further explore potentially effective drugs targeting these key genes, 10 groups of potential targeted drugs of the gene set were first obtained from the drug-gene interaction database. These drugs have been preliminarily explored in some neurodegenerative diseases, but have not been effectively explored in glaucoma, suggesting that these drugs are likely to become effective clinical candidates for the treatment of glaucoma. To explore the affinity and mode of action between drugs and proteins, we screened 4 groups of drug-protein complexes with lower binding energy and higher affinity by molecular docking technology. To explore the specific expression and distribution of core genes in the cells of the TM tissues from glaucoma patients, we first clustered the TM tissues into 17 categories by analyzing the single-cell transcriptome data of the TM tissues, which were further annotated into 6 different cells. The expression profiles of the 5 core genes in different cells of the TM tissue were investigated. These results suggest that these core genes may play an important synergistic pathogenic mechanism through immune cells and trabecular meshwork cells in the trabecular meshwork of POAG patients. For the first time, we explored the association between SLE and POAG based on gene transcriptome analysis. Through bioinformatics analysis, we identified some common markers and pathways between the two diseases. Meanwhile, for the first time, we explored the correlation between the pathogenesis of these two diseases from a new perspective. We also performed experimental verification of diagnostic biomarkers and targeted drug prediction for these two diseases, hoping to provide scientific basis for future research.